you. So I'll start by saying thanks to everybody for coming to the workshop. Seems like it's going pretty well so far, and it's been really great to work with this group of people for the last couple years. Excuse the gimmicky title, A Tale of Two Exudates, but it's pretty much what I've been thinking about for the last couple years on this project. But more specifically, the role of citrate and phytase in unlocking the potential of soil organic phosphorus for plant nutrition. So some of these issues may have already come up in your breakout group sessions, but what fascinates me is how large of an impact humans have had on the redistribution of phosphorus globally. So you have these regions of intense phosphorus surplus and regions of deficit where there's not enough phosphorus for crop productivity. And so I'm very interested in phosphorus use efficiency in agricultural systems and how the imbalance of phosphorus in soils can lead to really bad environmental impacts like aquatic ecosystems. So the broader question is, how can we improve the use of phosphorus in agricultural systems? And so the last couple days we've been hearing about ranges of organic phosphorus pool size in soils, and that can change depending on where you are, what soil you're looking at. What we do know is that in most soils, the available pool of phosphorus is very small. So this is mostly soluble and inorganic forms of phosphorus, and the trick is getting more of this organic pool into that slice of the pie. So how can we do this? I'm interested in rhizoengineering for phosphorus. So this is the idea that you can modify the rhizosphere in a way that promotes both the solubilization and mineralization of organic phosphorus so that more of that stable organic phosphorus pool can be mobilized to the plant. And we know from test tube experiments and soil batch experiments that if you add an organic anion to the solution, you can improve solubilization of both inorganic and organic forms. And if you then add a phosphatase enzyme, such as a phytase, to that mix, then you can release a lot of available inorganic phosphate. Now, in a plant system, it's much more complicated because basically you need both of those processes going on and producing soluble phosphate near the plant root where the plant can utilize it. So there's a spatial effect here, but there's also a large microbial effect, which is that these exudates may also be influencing microbial function and community structure. But more specifically, what I'll be talking to you about today are some of the first couple plant experiments we did with the transgenic plant lines to look at the interaction of citrate and phytase in the rhizosphere and see how that impacts on phosphorus availability. So how can we do this? Well, I've had the opportunity to work with a collection of transgenic plant lines that were developed at CSIRO by Alan Richardson and Tim George. And these include a wild-type tobacco plant and also a set of transgenic plant lines. So we always have a transgenic control. This is one that's undergone the plant cell culture process, and so it doesn't actually include the gene for the trait that will be expressed in the plant, but basically it's the control for most of these experiments. We also have single exudate lines. So these are ones that are constitutively expressing a citrate transporter from Arabidopsis or a fungal phytase from either Aspergillus or Pinophoria. And there's ways that you can combine these exudates into one plant, either by crossing those traits into one plant line, or you can grow one of each plant in the same pot, which we call the intercropped condition. So the questions to ask with these are, first, does the combination of citrate and phytase improve soil P availability? And then following on from that, what is the spatial effect? Do you actually need root intermingling for a complementary interaction between these exudates to occur? So these are the two questions I'll address today. So in the first study, this was published earlier this year in Plant and Soil, and it asked that first question. But we looked at a shoot pea accumulation here and all of the plant lines and combinations that I just described. We saw that, yes, indeed, when you combine these traits in the same plant system, then you do see an improvement in shoot pea accumulation compared to the single phytase or single citrate line. And you also see that, so this is the crossed condition here in the purple bars, and where you have the plants growing together in the same in the same pot. We also looked at changes in extractable phosphorus pools in the soils around the roots of these plants. And this data is based on a citrate extract, and we decided to go with citrate extract because there was a study of 32 soils in the UK led by Mark Stutter, and they found a really strong correlation between citrate extractable phosphorus and monoester phosphorus determined by NMR. So we thought this would be a better way to look specifically at that pool of phosphorus that might be affected by these traits, and also even look at phytase labile phosphorus in those extracts. So you can see here is that there's not a huge change in organic phosphorus in the citrate extracts, and you see a larger variation in the blue portions of the bars, which are the organic fraction, but it's actually in the treatments where both the traits are combined, you see the biggest depletion of the phytase labile fraction of these extracts. So in this first question, we were able to show that yes, when you combine these traits, either by crossing those traits into the same plant or by growing two plants in the same pot, you can see improved shoot phosphorus accumulation and also increased depletion of specific pools of organic phosphorus. What was interesting from this is that when you actually looked at complementarity, there was a bit of a difference between whether you grew two single plant lines together in the same pot or had those traits being expressed from the same root. And to show this, this is the land equivalent ratio, so this is a metric for complementarity, and it basically says that, well, if a plant grown in an intercropped treatment is accumulating more phosphorus than a plant grown with itself in a monoculture, then you'll have a value greater than 1. And so we saw in both of these dual trait treatments, you see values greater than 1. But what's interesting is that where the exudates are being produced from the same root, the effect is even larger, which suggests that there may be some spatial component to this, that in fact the effect of having both citrate and phytase produced from the same root is much greater than plants that need to basically grow towards each other before those exudates can interact. And to add to that, in the cross plant lines, the exudation of citrate and phytase are actually about half of the single plant lines, which means potentially that getting these exudates together in the right spot at the right time, close to the root, could have much larger impacts than we're actually able to measure in this study. So the second question was whether root intermingling would be required to produce this complementary effect. And to test this, we did a root intermingling and root exclusion experiment alongside. So we had the citrate, a single citrate plant line, and a single phytase exuding plant line growing together in the same pot, either with their roots allowed to intermingle or separated by a 20 micron mesh barrier. And here I'm showing again this land equivalent ratio, so any values greater than 1 indicate a positive effect of putting these plants together. Now in this, in addition to just citrate to phytase plant line comparisons, we included two citrate lines, one with a relatively low exudation rate of citrate and one that had about twice as much citrate efflux. We also looked at two different sources of fungal phytase, phytase from Aspergillus niger and phytase from Pinophoria lysii. And the difference between these is important because Pinophoria lysii phytase is considered to be more mobile in soils at this pH. So here we're testing the effect of citrate efflux, and then here potentially enzyme mobility, and seeing along the way whether you need root intermingling to see any positive interaction between these two exudates. So let me go back. Basically, this first one is showing that indeed, so this is the intermingled condition, and when you remove the ability of those plants to interact, then complementarity drops off. So in this case, where we have the plant expressing the A niger phytase and the high level of citrate efflux, you do see that root intermingling is required. What was interesting is that when you look at that same condition, but with the more mobile phytase, you see that it doesn't really matter that the plants are separated. And this is a permeable mesh. So even that tiny barrier was not enough for that enzyme to potentially move through and interact with the other plant, produce that benefit. So potentially there's some enzyme mobility effects here. And in terms of citrate efflux, so for some numbers, this is the rate of citrate efflux, nanomoles per gram of root per day, and then the higher rate. And what you see is that only at the higher rate of citrate efflux do you see these effects. But half that, and nothing really happens when you put these plants together. So potentially a threshold there for how much organic anion needs to be pumped out into the rhizosphere to provide a benefit to another plant with phosphatase production. So some of the factors that we learned about from these studies is that enzyme mobility could have a large impact on how these two exudates interact. So the distribution of enzymes in the rhizosphere is very important, and also where they're produced. Organic anion efflux, both the magnitude of organic anion efflux and also the longevity of those organic anions once they're in the rhizosphere, are very important factors here. And this kind of feeds into the microbial side of the equation, because we know that things like citrate and other organic acids are very labile carbon forms. So the effective concentration of how much citrate is actually in the rhizosphere is kind of an open question. And it may be that in that one condition where we saw complementarity with the higher citrate effluxing line, that there's just enough to both feed the microbes and promote phosphorus solubility, but we don't know. So now I'm interested in the effect of these exudates on a number of things. So I've been working with the assay developed by Marie Spong on the spatial distribution of phosphatase activity in the rhizosphere. So this is just a first look at the Aspergillus niger exuding plant line and the Pinophoria lychee exuding plant line. We're starting to get some hints here that phosphatase activity may be more dispersed in the plant line that produces the more mobile phytase, so that's encouraging. I also am looking into microbial response to these, both by working with folks at University of Guelph on phosphatase gene abundance and bacterial community structures. And of course, there's a curiosity about what these exudates are actually doing to the composition of phosphorus in the rhizosphere, and looking at some of that with NMR now. So there's much more detail on both of those things in the two posters. So I've got one there in the back looking at kind of the microbial side of how these exudates are impacting the rhizosphere, and in the back corner kind of a gorilla poster that I brought in without an abstract about the use of soil zymography. Thank you.